Can animal longevity studies truly translate to humans? And where does human research currently stand in this field? My name is Deborah, and today we'll explore the latest insights from longevity studies in animals and humans. But before we delve into the journey of longevity, don't forget to subscribe to our Healthnews channel and check the link below for our recommendations of top longevity products. Longevity studies exist in both animals and humans, yet distinguishing between the two is crucial. Animal studies provide valuable insights into the basic mechanisms of aging and potential ways to slow it down. There are far fewer longevity studies in humans, and most of them only focus on what could improve overall health rather than extend lifespan. So the question is, can animal studies translate to humans as well? Scientists have done a lot of research on animals to determine the mechanisms of aging. Even though many potential mechanisms have been revealed, they don't always translate well to humans. This is due to physiological differences between lab animals and humans, and many other reasons which we'll explore in this video. So you might be thinking, why don't we study longevity in humans more? Studying humans comes with practical challenges that don't affect animal research as much. Plus, ethical issues are a big deal in both animal and human studies, but they have many different aspects when it comes to studying humans. So let's explore these challenges in more detail. First of all, humans have much longer lifespans compared to typical laboratory animals used in research, such as fruit flies or mice. This means that studying longevity in humans needs long-term follow-up studies over decades or even generations, making it more time and budget-consuming. In contrast, fruit flies only live up to 4 to 6 weeks, whereas the lifespan of mice is 1 to 3 years, which allows scientists to obtain results significantly faster. Second, it's difficult to predict whether a certain drug or regimen slows down aging in humans because we're more susceptible to environmental stressors than most laboratory animals. We're not saying that animals aren't exposed to environmental stress, but the conditions in a lab are usually strictly controlled. In real life, humans may be affected by different factors, such as smoking, poor diet, or sedentary lifestyle that can contribute to aging and may disrupt study results. In addition, conducting longevity studies in humans involves additional ethical considerations that may not apply to animal research. Scientists must obtain informed consent from participants, ensuring they are aware of the health risks involved. If unwanted side effects of treatments arise, researchers must ensure the well-being of participants. Finally, the FDA generally approves drugs for treating diseases, and aging itself is not considered a disease, despite being a risk factor for many conditions. As a result, it might be difficult to get longevity drugs approved in the future, unless studies focus on specific illnesses. Overall, human aging is a complex process, making it extremely difficult to study. Studying longevity in animals is easier, but what have the results taught us so far? And most importantly, can they be applied to humans as well? Although many animal studies show promising results in extending lifespan, their application in humans is limited due to genetic and physiological differences, as well as environmental factors. But why are animals usually involved in longevity research, and what are the results? To begin with, most animals selected for longevity studies are species that we know well. The ones that are used for lab experiments are called model organisms and include animals such as fruit flies, roundworms, and mice. Besides being used to study diseases and behavior, they are also great subjects for understanding longevity. First, we know their full genome, which allows scientists to observe the expression of genes associated with aging. Second, these model organisms have short lifespans, which allows researchers to observe animals throughout their whole life. Finally, researching animals is much more cost-effective than doing similar experiments on humans. But remember, despite some genetic similarities with humans, these animals still have very different physiology from us. So take these study results with a grain of salt. Scientists have studied many ways to extend lifespan in model organisms, such as testing different drugs, restricting calorie intake, and manipulating gene expression. A lot of these studies have been successful in increasing longevity. For example, different studies have found that low caloric intake can lengthen the lifespan for roundworms even up to 50%. Furthermore, compounds like spermidine and NMN have also shown promising results. For example, according to a 2016 study, mice that received lifelong spermidine supplementation had increased lifespans compared to the control group. Based on research, it's due to spermidine potentially stimulating autophagy, a process responsible for getting rid of cellular waste. Similarly, it was found NMN is highly safe in mice and could extend lifespan by about 29%. The compound is associated with reducing inflammation, counteracting DNA damage, and promoting energy production in cells. So these results are very optimistic. But do they truly matter outside the laboratory? Although many animal studies show promising results in extending lifespan, their application in humans is limited. Despite these claims about lifespan expansion in animal studies, maybe we shouldn't get too excited yet. Some studies suggest that longevity methods that are effective in fruit flies might not be effective in mice and vice versa. Therefore, it's very questionable whether these studies could be translated to humans, considering we are more complex organisms. So, where does longevity currently stand in human research? Despite the claims made in animal studies, longevity research in humans is very limited. So, we talked about calorie restriction, spermidine, and NMN in animal studies, but what does human research have to say about it? First, just like in animals, calorie restriction might have the potential to increase longevity in humans. According to a 2023 study, reducing daily caloric intake by a quarter could potentially slow down aging. However, keep in mind that this is a short study that only lasted 2 years, whereas animal studies examine the entire lifespan. In terms of potential anti-aging compounds, spermidine has been a compound of interest for years. According to a study from 2018, higher spermidine intake was linked to lower mortality in adults. However, more studies are needed to confirm its safety and efficiency in slowing down aging. Larger and long-term studies of spermidine haven't been conducted yet and would likely require enormous funding. Similarly, NMN is yet another molecule of interest in longevity studies. NMN, often called the youth molecule, may benefit the body by counteracting many cellular processes associated with aging. For example, according to a 2022 clinical trial, NMN was shown to increase NAD+ levels in older adults, which may be linked to anti-aging effects and higher energy levels. However, just like with spermidine, more comprehensive studies are needed. So, after exploring the same longevity methods in animals and humans, it's clear that most longevity studies in humans are still inconclusive and in the early stages. The results in humans are not as optimistic as in animal studies, so it's probably too early to get excited just yet. However, there are still many ways to reduce the risk of aging associated with health issues. Keeping a healthy diet, staying physically active, avoiding smoking, and getting regular medical checkups are some of the many ways that may improve your health span. If today's deep dive has sparked your interest in longevity, don't forget to join our Healthnews community. Click the subscribe button for more science-backed videos about living a longer, healthier life.